I'm Michael Keller. I'm one of the team leads, and let me introduce the other three. Alex Konings, that's Alex. Christian Frankenberg, there's Christian, and Sasan Sachi. So we'll be coordinating the workshop this week, and you heard that we did a lot of work. I hope that it pays off. And my other job this morning is to introduce the short course. This is the short course title. I don't need to read it to you. You're seeing it for quite some time. And then I need to introduce the speakers. And you'll probably see me tripping through all the slides this morning because Michelle set this up in a very sophisticated manner that's difficult for a mere geoscientist. Our first speaker this morning will be speaking on the theoretical basis of plant hydraulics. Her name is Ashley Matheny. She's assistant professor at the Jackson School of Geosciences, the University of Texas. She has a PhD in civil engineering, so she's not a real geoscientist. And she specializes in ecohydrology, effects of drought, disturbance, and vegetation climate feedbacks. And with that, I call Ashley.
All right. Hi, everyone. I'm Ashley, and I am honored to be with you guys today. It is so cool to be presenting for all of you, mostly because as I go through this presentation, you're going to see a lot of your own work because they asked me to present on the state of the science and where we understand plant hydraulics and hydraulic theory. So this is me. Thanks, Michael, for the introduction. So I guess I'll start with why we're all here. And I think we all understand that we are here because vegetation is integral for the role it plays in the water cycle, recycling about 63,000 cubic kilometers of water every year, and also providing the largest terrestrial carbon sink. And so in terms of vegetation, when I think about it, that's what I think about for plants. But they also play the role of coupling below ground systems with above ground systems, transporting water back up into the atmosphere from the subsurface and occasionally the very deep subsurface, and their role in nutrient cycles. And then of course they're responsible for re-wetting the boundary layer, which leads to cloud formation and precipitation. And it just continues to play such an important role in so many different cycles. It's incredibly important that we understand it. And so the other cycle that we think about is often the energy cycle. And so our vegetation, as it's transpiring and taking up carbon, water is responsible for latent heat exchange in the atmosphere. And so that's the heat that's required to evaporate the water. And that plays an essential role in our energy budget balance. So whether that heat that's provided from the sun, whether that goes into heating up the air, which provides sensible heat, whether it heats up the ground or whether it evaporates water. And so when we think about plants and plant hydraulics, we're really touching on all of these things, on water, on carbon, on nutrients, and on energy, in a way that we can't decouple them from one another. And so when I started this, like Michael said, I'm an engineer and not a biologist. Was kind of taken aback by, okay, you want me to study water in trees? Well, how much water is really flowing? And so 63,000 cubic kilometers is a lot of water to think about. And so this video you're seeing is water that's leaking out of a birch tree. And it's not shaking because it's sped up. It's shaking because this is early spring in Michigan. And I'm shivering while I'm filming this video. And so this kind of gives you a feel for how much water is moving through that tree, how quickly. And if on order of scale, that blue wire that's wrapped around the tree is an Ethernet wire, like what you're plugging your household internet in with. And so there really is quite a large volume of water that's moving very quickly. And the way this water is moving is through the vascular system of the plants. And so our vascular system extends from the roots all the way up to the leaves. And when we think about our vascular system, the easiest way that I like to explain to my students to conceptualize it is to think about xylem as a series of pipes or conduits that are transporting the water from the roots to the leaves. And those vessels can be arranged in different ways depending on what species and what type of wood you have. So there's an example of ring porous xylem at the top and diffuse porous xylem at the bottom. And so trees are able to move their water in a way that they can control through regulations in their own structure, which take place over generations of trees, but also quickly through stomata, which are little pores on the undersides of leaves that open and close dynamically to regulate water exchange. And so when you think about water that's moving inside of the plant, you can start to think about it as a water tower. And so if you think about a tree that's had all night of not photosynthesizing, to recharge its vessels with water, it starts out hydrostatic equilibrium at everything blue. And then as the day goes on and transpiration happens, water starts to leave the tree through the ends of the branches first, and you start to experience these water stress gradients or water gradients where water is leaving, and then in the evening when transpiration slows down and ceases, the tree is able to replenish itself full of water and return to hydrostatic equilibrium. Hypothetically, but it doesn't always work that way, because occasionally these pressure gradients that are developing in the tree that are forcing the water to move become so steep that that water column inside of the xylem vessel can break or cavitate or embolize. And so when I tell my students to think about this, I have them imagine people playing tug of war, and that tension on the rope where those two teams are pulling becomes so much that it causes the rope to break. And that's essentially what's happening inside of the xylem system. And so Tim Brodrup really recently, 2015, developed a new way to look at embolism inside of plants visually. And so he was able to show what an embolism looks like as it happens. And so if this picture is our whole leaf venation structure, this is one embolism event that started in the midrib and was transported into the secondary and tertiary veins in the leaf. And then through time passes and it overlaps with a stack of images, he was able to demonstrate the pressures at which different vessels inside of the plant cavitate over time, and demonstrate clearly there are multiple cavitations taking place in the same vessels. And so as plants are functioning, as they have their stomata open for photosynthesis and transpiration, what they're really trying to do is take up enough carbon so that they're able to survive and have enough to eat, while not allowing this to occur in the hydraulic system. And so when we think about that as the hydraulic system overall, we often think about pressure volume curves or our percent loss in cavitation with respect to the water potential gradient. And so different plants experience water potential differences differently. Some of them are very tolerant of this hydraulic stress or large hydraulic gradients developing, like Juniperus virginiana on the far side, and others are not so tolerant of large hydraulic gradients, like the Quercus falcata that's closest to me. And so what Patrick is going to talk about to you later today, we'll define a little bit now, is that we can characterize the way plants are vulnerable to cavitation in terms of the potential at which they lose 50% of their hydraulic conductivity and the potential at which they lose 88% of their hydraulic conductivity. So once you've lost about 88% of your conductivity, you're pretty much going to stop your function. And so there have been theories that we as the scientific community have put forward about trade-offs within the plant system about, can you be both safe from cavitation and also transport water efficiently, and how are these things interrelated? And so in terms of this safety efficiency trade-off, the basic theory behind this is that if you have big vessels, like those ring porous xylem vessels that are very large and you're very capable of transporting a lot of water quickly, do you sacrifice being safe from embolism? And those vessels are frequently the first to embolize. But if you have smaller vessels that are more resistant to embolism, are you not as able to transport water quite as well as those big vessels? And so that's the essence of the safety efficiency trade-off. And so from this theory, we saw that from Manzoni's work that there was a specific xylem relationship for hydraulic conductance to that maximum potential gradient when transpiration is at its highest, that he found to be fairly well conserved across biomes. But in a recent study by Gleason, we found that there's not exactly a perfect trade-off between safety or efficiency. You're not choosing one strategy or another, because there are so many compounding variables that come into play. And we started this talk by talking about those compounding variables of all the different relationships that plants have with water and carbon and nutrients. And so when we think about these other relationships that are dictating the way plants move water, probably the first to be thought about is the way that stomata are operating. And so this is work from Bill Anderegg that was published in Ecology Letters last year, looking at how stomata maximize their efficiency. And so the theory has been, or a long-standing theory has been, that stomata operate to maximize water use efficiency. And so when I say water use efficiency, I mean the amount of carbon gain per unit of water loss. But the theory that Bill and his team put forward was that maybe instead of just simply looking at units of carbon and units of water, it's rather a risk trade-off. And so this trade-off that he put forward about the maximum xylem tension efficiency is essentially saying that rather than simply maximizing carbon per unit of water, it's how much carbon can I gain, or how much carbon am I willing to sacrifice gaining to not incur a risk of loss in the hydraulic system. And so in this sense, we find broad support for this new theory that carbon is maximized with respect to risk to the xylem system. And so when we talk about this and we talk about plant hydraulic theory, it has real implications for how trees and ecosystems perform. And so if you will take a trip with me to my field site in northern Michigan, let's talk a little bit about the consequences of these different hydraulic strategies and the different ways that plants operate their conductive system. And so this is a really classic example of two species that use opposing hydraulic strategies. And so this is my typical field site in northern Michigan. It's right up at the tip of the mitt where the upper peninsula meets the lower peninsula. And these two species, oak and maple, are now the dominant species in that forest. And so the oak and maple, if you've seen them, you know they have roughly the same size leaves. They support the same amount of leaf area. The trees grow to be the same height, and they have roughly the same size trunks. But when we experience soil water limitation, and so I will call this the mini drought because northern Michigan very rarely experiences an actual drought, we see that the two species respond very differently in terms of water flow in the xylem system. And so sap flow is a measurement. That's the sensor that I was installing when I was shivering filming the birch leaking. But it's a measurement that we measure at individual trees, and we frequently replicate it a lot. So generally 10 to 12 trees per species, and this is an average of all of those trees. And what we see is that our oaks will respond or not respond to soil water limitations. So as the cyan line in the top panel drops down, oaks don't really care about it, but our maples, the red line in the bottom panel, they strongly taper off their transpiration and hydraulic functioning. And so, of course, this wants to be asked the question of, well, why do these two trees that grow in the same ecosystem, that experience the same environmental forcing conditions and that look functionally the same, why do they do such radically different things? And so we can talk about that from all of the different levels within the xylem system. And we'll start at the top and work our way down to the roots. And so we started by analyzing the leaves and the way those leaves change their stomatal conductance in response to soil moisture. And the way we typically do this is through measurements that we make with a device that's called a pressure bomb or a pressure chamber. And we go up in a canopy lift and we measure individual leaves from the same individual tree or several trees of the same species. And we can construct these relationships for how the water content in the leaves changes over the course of the day. And so if we look at our pre-dawn, that should be the time when the tree is as charged as it's ever going to be with water before transpiration starts. And our midday, which should be the most stressed out part of the day, water potentials, we see different relationships with the red oak and the red maple. And so our red maple, on the side that's closest to me, while its early morning leaf water potential is strongly correlated with what the soil is doing, it's not so much in the afternoon. And it's striving to maintain that constant leaf water potential. And so traditionally, we dub this as isohydric, meaning that it maintains the same water potential. Our red oak, on the other hand, those blue dots, show no correlation with soil moisture in the morning, meaning that they're always able to refill their leaves, regardless of what the shallow soil moisture is doing. But in the afternoon, they experience lots of water loss in their leaves, and we call this anisohydric behavior, in that they're willing to incur a very steep hydraulic gradient to continue transpiring, to continue photosynthesizing. And so, if we talk about the ways that these species are different over the course of the day, the maple is sort of curtailing its function in order to maintain its hydraulic system to stay safe, and the oak continue to function to gain carbon. And we also see that these two species differ in terms of what they're doing inside of their xylem vessels. And so this is data that's collected with retrofitted soil moisture probes that have been stuck into the trunk of the tree to complement the sap flux measurements. And so those measurements, instead of measuring the velocity of water that's going through the system, are measuring the total water that's there. And so there's one sensor that's located about 1/2 meter off the ground, and there's another sensor that's located about 10 meters off the ground, right at the base of the live crown. And so for these sensors, what we're able to see is that our maple are emptying and refilling their vessels throughout the course of the day. And they're experiencing these large peaks and valleys in their amplitude of water content. So each day is an up and down fluctuation. But then as the soil moisture, as the black line, starts to go down, our red maple are less well able to replenish their internal capacitance or their internal water supply. And they stop functioning. But our oak really only experience those great big diurnal shifts when the soil moisture limitation becomes extreme. So their amplitude increases as you go towards the far side of the screen. And so while this is interesting on its face, it gives us another metric to look at how plants experience stress and when the onset of stress occurs. And so if we look just at that red maple, because the oaks didn't experience stress during this condition, we find that there's a really sharp bifurcation in maple's behavior of how it's replenishing its stem water content based on what soil water potential it exists at. And so as the soil gets dry, we see this very strong linear relationship of how much water is being withdrawn each day, whereas there's not really a steep relationship at all, or any relationship, when the soil is wet. And so if we're able to identify this threshold of stress-induced response, then we can start to talk a little bit more about how the plant functions during wet and dry conditions. But if you've been thinking about those leaves and you've been thinking about the xylem and the strong relationship between maple water flux and oak water flux with respect to soil water content, you're probably thinking about roots by now, and the difference between the two trees being their rooting structures. And so we used deuterium excess. So we extracted xylem water cores and we did isotopic analysis of the water. And this deuterium excess parameter exists, and that the isotopologues of water will evaporate differently based on their weights. And so we were able to show that our water that is contained in the xylem of oaks is less evaporated than the water that's contained in the xylem of maples, indicating that our oaks are taking up water from a deeper source that's less able to be evaporated, so not the shallow soil layers, than our maple. And we also see that the maple water isotopes vary strongly through the course of the season, and they track what precipitation looks like. And so really, if we think about this, our oaks and our maples, those two species that exist in the same place and they look functionally the same, differ at every level in terms of what they do in terms of their leaf water regulation, in terms of water content in the xylem system, and in terms of where they get their water. And so this really gives us a framework for understanding plant hydraulics from a whole plant perspective, rather than just talking about what one individual organ does versus another. And so this is a paper that was put forward by Kate McCullough's group, and out of this review came this beautiful diagram of everything that comes together that influences a tree's hydraulic strategy. So starting with roots and rooting depth, but also understanding that soil and the environment that the tree is growing in will play a key role in how those roots develop. So if it's super rocky soil where they're rooting through fractured bedrock, or if it's glacial soil where they're able to root as deeply as they want, how that influences root dynamics and root morphology, how woody habit influences their stem capacitance. So is there a relationship between the density of the wood and its vessel size and how well those vessels are able to transport the water? And then, of course, within the leaves, thinking about it in terms of water demand and water supply and how those leaves are regulated, whether they want to keep their water or whether they allow those steep water potential gradients to develop. And so as we think more about that concept of isohydry, because this is going to be a word that we talk about a lot over the course of this week, I think, it's important that we think about it as a whole plant response. And so this is a figure that was created by Jordi Martinez Villalta, relating the way all of those signals come together to influence the water content in the leaves. And so this is sort of flipped upside down. Our leaf water content is down here in the bottom, and our soil water potential is up at the top. And so from this figure, what we're looking at is when the soil water content changes and, say, becomes more limiting, our plant hydraulic conductance, or that KH, will decline, because that KH is related to the amount of water that's there, and will also experience a decline in leaf conductance. And those two things will have opposite behaviors. So leaf conductance will reduce the gradient of water stress within the xylem system, but a loss of hydraulic conductance will make that gradient steeper. And all of these things, depending on how the plant will balance one against the other, will combine to influence the leaf water potential. And so Kim Novick took this one step further And she extrapolated this to the apparent sensitivity of our leaf water potential to soil, but also with respect to what the leaves are experiencing in terms of their environmental condition. How is the vapor pressure deficit influencing the water inside of the leaf? And that vapor pressure deficit is also being influenced by the amount of water that's available in the soil. And so in the same vein, she's thinking about the way that leaf water potential responds to leaf area. So many trees exhibit a phenomenon called drought deciduousness, where when drought stress becomes too extreme, they will drop their leaves. And so as soil water can decline, leaf area can also change, and that will change the amount of water that's available to the remaining leaves on the tree, which will play a role in their leaf water content. And in this sense, we've also presented the difference between our leaf water relationship with soil water content and the stomatal relationship with soil water content. And so these two things are becoming decoupled, but we're still thinking about it in terms of a whole plant response rather than just simply what an individual leaf is doing. And so work from Stefan Meinzer's group has used leaf water potential differences between midday and pre-dawn. So midday in this case is your y-axis, so that would be our minimum leaf water potential we experience. And our pre-dawn is our x-axis, and has characterized hydroscapes. And so in this case, because we're just looking at pre-dawn and midday, this is integrating together all of those things that influence leaf water content and not necessarily talking about stomatal regulation strategy, and is defining what he calls a hydroscape, or the area that exists between the one-to-one relationship and where that plant actually behaves. And so in this sense, our anisohydric species, those species that will allow their water content to vary greatly depending on what the environmental conditions they're experiencing are, have large hydroscape areas, or that big red triangle. And other species that are more isohydric will have smaller hydroscape areas. And so when we think about how we classify plants in terms of hydraulic strategy and the way they function, one of the things that we think about a lot is the way that hydraulic traits work together to facilitate these strategies. And so in recent work that came out of Fu, which is one of the folks in Meinzer's group, they were able to show that that hydroscape area is connected to stem capacitance and leaf capacitance and leaf and stem hydraulic conductance and conductivity in significant fashion. But they only showed 6 species. And so as we start thinking about this, and for me, 6 species is good. When I work in northern Michigan, I have about 6 species total that I worry about. When a lot of you folks work in your field sites, you're dealing with the tropics, and you do not have 6 species. And so as we think about the ways we can characterize trees and tree hydraulics at large scales, we like to look to our ecologist friends who do wonderful jobs at putting together large trait databases and try to connect how things function on a global scale. And so this is work that came out of one of my undergraduate students, trying to use the Global Plant Trait Database to look at the way traits like rooting depth, isohydricity, and wood density come together. And one of the things that we really struggled with when we were putting this paper together is that not all of these traits are recorded for all of the same species. And even then, when we start talking about things like rooting depth, it's very hard to know for sure. And when we start talking about things like isohydry, or even our vulnerability curves to cavitation, those curves that I told you that Patrick is going to talk a little bit more about, when we start to talk about those things, it's important to understand whether those measurements were made on an individual leaf, whether it was a sun leaf or a shade leaf, where you existed within that species range. If you were measuring that pressure volume curve on a leaf or a branch, and if you were measuring it on a branch, was it a distal branch, was it a large branch, and how was it measured? And so these relationships are often very spurious and difficult to measure. And the one that I'd like to draw your attention to most is about roots, that it's not just about rooting depth. So all of these figures that my colleague Liz has developed, these are for the same root length density, meaning the same length of root per unit volume of soil, just arranged differently and partitioned between whether it exists as a coarse root or a fine root. And so depending on how you look at this structure, you could have the same plant trait parameter that would parameterize your roots while having what exists as a totally different morphology in the field. And so when we characterize these things and talk about hydraulic strategies and hydraulic traits and trait relationships, it's important that we get them right. And it's important that we develop a theoretical understanding for them well, because when they come together, they will affect the way our ecosystems function. And so if you think back to my field site and our data from the red oak and red maple, where the red oak really didn't care what the shallow soil was doing because they have access to deep water, and our red maple were strongly related, we see that that relationship continues into bole growth. That if we were to look at how those trees develop over many years, and so this was 14 years of data collected for more than 100 trees of each species, that we see that our maples are really strongly related to what the soil moisture is doing, and our oaks aren't. And so when we talk about ecosystems and ecosystems functioning going forward in time and in climate, it's very important that we be able to characterize these different responses. And to take this one step further, most of you know that that field site in northern Michigan is paired with a disturbance experiment. And in that disturbance experiment, we artificially accelerated the succession of the landscape by killing half of the trees in the forest by stem girdling. And that experiment took place long enough ago that the forest has recovered in terms of the total amount of leaf area. And the real difference that remains between those two systems now is what species that leaf area is attached to. And so whenever we look at our disturbed site, most of the leaf area exists on the oaks and maples and some beech and pine. And in the control forest, they're still pretty evenly distributed, but aspen makes up a very big component. And so when we look at how those two sites perform at an ecosystem level, so using eddy flux data, which is representative of several square kilometers of forest, what we see is that during wet conditions, when plant hydraulics really aren't playing a huge role, when everyone has as much water as they could possibly want, the two forests perform similarly, almost exactly the same. But when soil moisture becomes limiting, and the soil in our site is sand, so soil water contents less than 4% are pretty typical for us for those dry interstorm periods, we experience upwards of 25% difference between how those two forests function, because those differences in plant hydraulics are playing a strong role in regulating what each tree does, which changes the way the forest as a whole performs. And so this is great and all, but this is my case study. This is my field site. What makes this alarming is that these different strategies, coexisting species with different hydraulic strategies, are common throughout ecosystems and throughout the world. And so as we talk about the way our forest dynamics will change in response to drought and response to disturbance, it's not a unique problem to my site that you need to understand the plant hydraulic system. But as we move forward, thinking about that plant hydraulic system, how can we extrapolate from measurements that we make at a single tree, when I study sap flux or when I study the water content inside of the trunk of a tree, or when my REU students go out and they measure leaf water potential and branch water potential, how can we extrapolate those measurements to look at broader ecosystems at a regional or even global scale? And so that's where remote sensing comes in. And I promise, Alex, I will only talk for a second about remote sensing things. And so when we scale from trees to ecosystems, we can do it sometimes. And so we can do it through a combination of multispectral imagery, which allows us to break down a forest into its composite species, and we can use LiDAR imagery to give us the sizes of those trees in the forest. And we can bin things together and say, okay, we have this many large maples and this many small pines, and if we assume that they do functionally the same thing, we can talk about scaling from a tree to an ecosystem, and that works well with some measurements. So for example, sap flux measurements scale well from trees to ecosystems when you have a large enough sample size. But where this really becomes a problem is when you're not talking just about things that are sap flux, which is integrative of different traits, because that sap flow rate is going to be irrespective of what the roots do and what the leaves do and what the xylem looks like. But when you get down to the nitty-gritty of measuring a pressure volume curve or measuring leaf water potential and tracking it over the course of the day or over the course of a year, things become much more challenging to scale. And so this was another paper from Bill Anderegg's group looking at the way things change within a species. And so even within a species, you'll have differences from leaf to leaf and branch to branch, depending on where that branch is, whether it's a sun branch or a shade branch. You'll have differences among individuals within the same plot, within the same site. And then you'll have differences based off of whether you're in the northernmost extents of the range in Alaska or whether you're almost down in West Virginia in the southern extent. Because those trees are experiencing different subsurface conditions, they're rooting over different geologies. They have different environmental conditions that are influencing the forcing of the leaves. And then even if we just look at one tree and we track it over the course of its lifetime, what it does as a sapling and the way it develops will be different. And so its pressure volume curve will change as time goes on. And so depending on that life history of the individual that you're measuring, what we're thinking about in terms of plant traits aren't static. And so the other thing, whenever the directors contacted us and they asked us to give these informative lectures for you using your own research, is they asked us to talk about the most important new directions for where the field is going. And so when you think about that tree that experiences a drought, it maybe doesn't die. So it suffers, but it comes back, it makes it back. How does that change the way it functions? And so for me, one of the most interesting new directions that this field is taking is looking at our recovery response to drought. Do we have hysteresis in our plant traits as drought happens and as it comes back? And so in this figure, the pre-stress condition, those blue dots up at the top, you can't see my mouse, but I can see my mouse. Those pre-stress dots, it's okay, we've only got 4 minutes left, we can do this. But those blue dots, where it's experiencing a certain water potential, so maybe -1 megapascal, have significantly higher stomatal conductance than what that same plant will experience at that same water potential in its post-drought condition. And so we see this hysteresis in plant traits if we consider a pressure volume curve to be a trait. And so if we look a little bit closer, we can see that if that plant experiences a drought stress cycle over time, the way it responds will continue to change. And so in this figure, we go from first cycle in red to last cycle in blue. And because I'm a hydrologist and I'm not a geologist, what we're looking at is Q, so Q being our flow rate, and delta H is our hydraulic gradient. We see different responses where the reds almost start out as a plastic response, where they experience stress, they have some hydraulic gradient, and they're able to perform at a flow rate. But then as time goes on, and as drought stress continues to increase, we see bigger and bigger hysteretic loops. So for that same hydraulic head, you have 2 different flow rate conditions. And so as this plant is experiencing water stress over the course of its lifetime, the way it performs will not always be the same. And so when we talk about the future and future directions of the field of plant hydraulic theory and our understanding of plant hydraulics, not only do we need to be considering this as a whole plant response, but we need to start to look a little bit deeper in terms of how we understand cavitation and refilling and vessel response. And so this was really an exciting presentation for me, was Gabby Catul's talk at AGU last year, where he presented work from Murad. And what they were showing was thinking about xylem networks differently. Usually when we think about xylem, we think about porous media flow, and Pierre and I talk about Darcy's law and Richard's equation. But what Gabby's group was able to do was they were able to use the Young-Laplace equation and consider xylem as a more complicated pipe network rather than just porous media flow, and show the propagation of embolism within the system and the way xylem vessels were becoming isolated and how things changed. So for me, looking forward, these are a few of the things that I'm most curious about, is how do these things come together to tell us more about the way plants and forests will respond to drought when they're not 100% dying. And so because I've got the red light on, let's go ahead and we'll go to the acknowledgements, because science never happens in a vacuum, and all of this is mostly courtesy of you guys for providing the wonderful literature that I was able to bring in, but also to my team and my collaborators. And with that, we can get the discussion started if you'd like. am happy to take questions or field them to you all who did the research. So thank you.